While people are coming and still joining the room, I would like to welcome everyone. My name is Oliver Fien. I'm director of the West Coast Metabolomics Center. Again, welcome everyone, and special thanks for Dr. Ling to present today her work. Let me introduce her. So Dr. Ling is an assistant professor of chemistry at the George Washington University in Washington, D.C., with currently 1,400 citations on her papers. Her area of expertise is bioanalytical chemistry, mass spec, proteomics, metabolomics, and the application on neurodegenerative diseases. She started with a bachelor at China Agricultural University in 2012 in Tsinghua University, before she did a PhD at Professor Ling Lee's lab at University of Wisconsin-Madison in 2017. And then she did a postdoc at the NIH in Bethesda in 2019 and started her independent research group at George Washington University in 2019. Remarkably, she already has received a very prestigious NIH R01 grant to support her research in developing novel and improved bioanalytical methods using LCMS platforms and applying the combination of analytical chemistry, bioinformatics, and cell biology approaches to study human diseases. So she implements multidisciplinary approaches in human-induced pluripotent stem cells, or IPSCs, derived neurons, but also in mouse models and in clinical human subjects, to study the molecular mechanisms of neurodegenerative diseases. She has also been selected as an emerging investigator by the Journal of American Society for Mass Spec, with a research article featured on the journal cover. So I'm very pleased to welcome Dr. Ling, and the floor is yours.
Yeah, thank you, Oliver, for the really kind introduction. And it's actually very nice to see people on Zoom, especially we sort of met briefly at ASMS last week in person. So it's kind of nice that we're back to Zoom for meeting. So I'm going to start sharing my screen. Yeah, so today I'm going to talk to you about how we develop mass spectrometry-based method to study the human brain, and specifically the neurodegenerative disease. So we know, a lot of them already know that neurodegeneration is about the progressive death of neurons in the human brain. So we know that a lot of genetic mutations can actually cause different kind of neurodegenerative disease, depending on where the location of that neurons located in a different region of the brain. So if the neurons started dying that controls the language, then patient have language barrier. If the neurons that started dying controls the motor and the function of the movement, then patient have motor neuron dysfunction and also start to have movement disorders. So those, because of the genetic mutation that could run from the family, could also be sporadic mutations that people pick up during the environment, or a lot of the pollutant or contaminations or working environment when people are getting old. So we now know that a lot of risk factors contribute to the neurodegenerative diseases. But what we don't know is how do those genetic mutations influence the molecular processes. And because of that, we don't really have any cure of neurodegenerative diseases. And actually, in general, most of the disorders about human brains are not really having a good treatment. So one of the direction that my lab wanted to contribute is to develop analytical platforms to help us understand the molecular processes underlying those molecular neurodegeneration. So just to summarize of what we already know in the field, so we know for degenerative neurons is obviously not healthy and is not happy. A few key factors contribute to the neurodegeneration of the neuron: lysosomal dysfunction, mitochondrial dysfunction, could be misfolded proteins started to aggregate and started to stress out the cells, and impaired RNA homeostasis, and also impaired neurotransmitter pathways. So we know that a lot of the genes could contribute to the dysfunction of those organelles, but the detailed process are not really well understood. So, and how do those gene mutations eventually lead to the neurodegeneration and unhealthy state of neurons are not known. So two of the organelles that my lab are really interested in are mitochondrial and lysosome. So we all know that mitochondria is a powerhouse for cells, right? It generates ATP, and our human brain occupies 2%, about 2% of the body weight, but it consumes 20% of the energy. So it really need, especially for brain health, really need to continuous generating those ATP. So mitochondrial health is extremely important for brain health. Lysosome, on the other hand, is more like a trash disposal system, where if you have, we generally generate different trashes, like everybody, and we generate trashes in a city. It's our cells, just like a city, they generate the trash a lot of the times, and then it needs to be recycled, and proteins need to be degraded into amino acids, so the cells can actually absorb them again and recycle them. So lysosome is this trash disposal system. So especially for neurons, which are post-mitotic, so they don't divide anymore, not like other skin cells or stem cells. They don't divide anymore. So if there's any insult, they can't really divide it to dilute those insults. So they highly rely on the lysosome health to be able to recycle and degrade those misfolded proteins. So you can imagine if the misfolded protein and protein aggregates couldn't be degraded by the lysosome, and it's going to start to stress out neurons in different regions of the brain. So that's what my lab are mostly interested in, to study mitochondrial and lysosomes. So today I'm going to talk about 3 major ongoing projects in the lab. First, relatively simple method development, like not much method development, but more of an application of different omics for biomarker discovery. And then I'll talk about how we develop organelle proximal labeling approach to study the dynamic and in vivo lysosome and mitochondrial interactions. And then I'll talk about how proteins turn over in neurons and how we can use that to understand the neurodegenerative processes. Okay, so starting from this first project about discover biomarkers. So when we think about neurodegeneration, we mostly think about, oh, it's only old people get it, but actually not true. So one of the disease called MELAS had a quite long name. The age of death is very young. So this is mostly because through the from genetic mutations, where if there are mutations in the mitochondrial DNA, we know that mitochondria also have DNA for itself. Those mutations can pass through the family, and this is maternal maternal inheritance, because we actually get most or not all of the mitochondria from our mother. So if the mother have mitochondrial DNA mutation, it will pass down to the children. And our mitochondrial is very interesting. It can also split itself and merge together. So if there are some of the mitochondrial mutations start to influence not all of the mitochondrial, but some of the mitochondrial. So if the dysfunction mitochondrial start to occupy the majority of the cell, then the human body will have different problems at different actually locations of the human body to have generating ATPs, or how the mitochondria is going to influence a different part of the organ. So this MELAS disease actually not only have symptoms in the brain like stroke, it also influences a lot of like kidney and liver diseases. So we know that mitochondrial DNA mutation definitely cause mitochondria to not be happy, and it influences the mitochondrial complex, OXPHOS complexes. But we don't know how the details of this process. So we look into this process through a collaboration with the Children's National Hospital in Washington, D.C., where MELAS is actually a really rare disease, because if the mitochondrial mutation is too severe, actually it might have birth defect. So we were able to find, we started a pilot study first, where we collected patient fibroblasts from MELAS patients, and we had the patient's mother as a healthy control. So it's a unique pair of control versus disease, because the mother passed down all the mitochondrial DNA to the daughter, but the mother is actually healthy. That's because majority of the mitochondria in her body are healthy mitochondria, where when they pass down to the daughter, some of the mitochondria that had mutations actually had a majority of them keep splitting, and that occupies the majority of the mitochondria. So in the patient, the daughter has actually very severe MELAS syndromes. So we cultured one of the reasons we wanted to choose patient fibroblasts for this biomarker study is because we usually do biomarker study use with like urine samples or blood samples or cerebrospinal fluid. But for mitochondrial disease, those biofluid doesn't have mitochondria. So if you look at mitochondrial diseases, you really wanted to look at inside the mitochondria what's happening. So that's why we decided to use the patient fibroblasts that and grow them in different biological replicates and look at that as a pilot study to look at what goes wrong with mitochondria. So in this case, we want to look at all kinds of molecules with mass spectrometry. So we did a traditional methanol water chloroform extraction, where we can get the polar molecules from the methanol water fraction. We can get the proteins in the middle, and we can get the non-polar metabolites and lipids from the bottom. So my suggestion actually to do multi-omics, especially for both small molecule and proteins, is if you have enough sample, we've done this before, that is actually better to separate the sample into two aliquots and do them separately. But for our case, the patient fibroblast that has this mitochondrial mutation doesn't really grow. So it's really hard to get a lot of material. So when you have limited material, then you can think about to use the same sample and then derive and then fraction that into different fractions for different type of molecule analysis. So when I first started my lab in 2019, I know I wanted to do both small molecule and protein. For the small molecule, the first thing I did is to build our in-house metabolite library. So we just keep continuing buying the different compounds. The reason that I did this is actually did a lot of metabolomics studies when I was still a graduate student. At that time, the data analysis was quite challenging because we don't have much really useful, really good online tools. And the library for the LCMS data is not also not as robust as nowadays. So the approach we did when I was a graduate student is we do all of the LCMS for all kinds of metabolites, and we don't look at identification first. We do statistical analysis first. Then we pick out those that are significant. And then we go to try to identify those statistical significant metabolites. So that really, I did a lot of manual work at that point. And then gradually nowadays, the online database is getting better. The platform is getting better. So when I first started in my lab, I think that I wanted to approach in a different direction where I wanted to first try to identify as many compounds as possible with a confident identification. And then we can look at the statistics at the same time with this metabolite identification. So the first thing I did actually is to buy a bunch of small molecule compounds and stock it in the lab and try to analyze them with both RP, HILIC, and different kind of mass spec, tandem mass spec to build our library for a confident metabolite identification. So in this sample, we also created a pooled aliquot using as a QC, so we can use it to monitor instrument stability, and also use that to assist our metabolite identification. So we do a lot of MS on those pooled samples and compare that with the library. So the advantages of doing this approach is we don't have to do DDA for the real sample, because although DDA data dependent acquisition, where we acquire both MS1 and MS2 at the same time, could save a lot of time for MS2, but we do found that the peak shape sometimes might be funky. So the way we're approaching this problem is we use the pooled sample for a lot of DDA and MS, but the real biological comparison, we're using just the full MS scan for all of the samples. And we were able to get a very complementary coverage of different type of molecules. Those are confident identifications using different approaches. And this is led by a graduate student in my lab, Haorong. And then we also want to look at the proteins in those molecules, in the patient fibroblasts. So the approach which took is we first conduct the DDA proteomics, global proteomics analysis. Then we validate those protein hits with DIA approach, with both the direct DIA software and the recently developed max DIA software using MaxQuant. So we see that they actually validated they were actually quite consistent. And a lot of the proteins we see belongs to complex 1 actually were downregulated, consistently downregulated in different kind of approaches. So then we wanted to combine them together into the same metabolic pathways. When we do the joint pathway analysis, we can see that the arginine biosynthesis and purine metabolism really stand out. And you can see that the proteins in boxes and the small molecules in the circle were actually can be identified at the same time. So I just wanted to zoom in on 2 of the most important, interesting aspect of those joint pathway analysis. One is this OXPHOS complex. So we actually know that its mutation specifically influenced the complex 1 of OXPHOS complex. So when we look at the proteins, we indeed find many complex 1 proteins are downregulated together with complex 4 proteins. But we also see complex 5 proteins, which is ADPase. A lot of them are upregulated. This is ATP synthetase. We think this is probably a compensation process happening at this point, and really trying to compensate for the ATP. Because when we do a seahorse assay for those patient fibroblast cells, to measure the mitochondrial respiratory activities, we do find a downregulation of ATP production in those patient fibroblasts. So this might be trying to compensate this ATP, but because of the complex 1, that a majority of them are so dramatically downregulated, the total ATPs produced by the fibroblasts are still dramatically reduced. Then another one that stands out is this arginine biosynthesis pathway, where you see this arginine is significantly downregulated, but the citrulline is actually upregulated. So citrulline is one of the molecules that contribute to the synthesis of arginine, so they actually recycle back. And so what we think is happening is the arginine synthetase enzyme is significantly downregulated. That causes the synthesis of the arginine pathway to be downregulated. So you have a downregulation of arginine, which actually contribute to a lot of the neural symptoms, stroke, and dysfunctions of the brain in the patients. And then the pathway is trying to compensate this arginine processes. So citrulline, so you're trying to upregulate this ASL and NOS enzyme, but it's still, because ASS1 is so much downregulated, it wasn't able to rescue this process. So you still have arginine deficiency. So one of the interesting things for this project is the patient actually have weekly dose of arginine infusion that they are going through. So, but even with that, when we take the patient's skin cells, we still see a very dysregulated arginine biosynthesis. So because of that, we actually are right now talking to the physicians that take care of the patients and patient families to suggest whether they can increase the dose of arginine infusion or the frequency of arginine infusion, so that we can really help the patients recover, or at least reduce some of the symptoms caused by the arginine deficiency. So that is one of the not much method development, but just like a proof of principle of pilot proteomics and metabolomics analysis. And the next ongoing work is to validate this arginine biosynthesis with a larger patient population. So this, especially I wanted to focus on that to validate whether we can use this ASS1 as a therapeutic target. So if we can really increase the regulation of this one, maybe we will be able to rescue some of the arginine deficiency in the patient brain. Okay. So so you might think, so we're looking at mitochondrial dysfunction from this pilot study, right? So you might probably ask this question: why don't we just isolate mitochondrial? So there are a lot of approaches that we can isolate mitochondrial. Mostly there are majority 2 approaches. One is you can tag the mitochondrial with an immunotag and then do a immunoprecipitation. Another way where most people do is do a centrifuge. So you do a sucrose centrifugation. So the problem for that one is it's not pure. It's really hard to get a pure isolation of mitochondrial. The second problem, especially for this mitochondrial dysfunction, is a lot of the mitochondrial, if they're not healthy, they tend to burst when you're trying to centrifuge them. So if you try to get a mitochondrial isolation, at the end, you're only getting those healthy mitochondrial because the unhappy mitochondria already burst and you get rid of the supernatant. So you can't really isolate mitochondrial if the mitochondria are not healthy. So then we wanted to think about, is there a way we can actually get those mitochondrial molecules, both small molecule and proteins, from an in vivo approach before this we kill the cells. So that comes up with the second major projects in the lab that we're working on, is to develop in vivo proximal labeling approaches so that we can actually tag those proteins and small molecules in the different locations of subcellular locations in the cells before we kill the cells so that we can pull them down easily. So I wanted to talk about the proximal labeling approach. This is one of the approach that developed not long ago, about 10 years now, by the Alice Sing's group, that we can actually use it to study the molecular microenvironment. So I'm going to talk to you about how it actually works. So this proximal labeling is happening in vivo, where we first need to tag an enzyme onto a protein of interest genetically. And then we can express this construct into the cell. There's a variety of different enzymes that you can tag to a target protein. It could be peroxidase, so that would be APAX and HRP, or it could be biotin ligase, it could be bio-ID or turbo ID method. So you first genetically tag this enzyme to a protein, and then you can express it in the living cell. In the cell, you can actually activate this enzyme. Different enzyme of proximal labeling will actually use different ways to activate this enzyme. So if you're using peroxidase-based proximal labeling approach, you would activate with a 1-minute treatment of H2O2, because it's a peroxidase, right? It will be activated by H2O2 and biotin phenol, which is the substrate that is gonna enzyme is gonna use. If it is a biotin ligase, you can actually you don't have to add ATP, because the cells in living cells generate ATP all the time. So you just need to add biotin to the cell. And then you can activate the enzyme. The enzyme is gonna create a very narrow 10 nanometer radius, and it's gonna biotinylate the surrounding proteins and all the other molecules surrounding this enzyme. And then you can actually lyse the cell and enrich those biotinylated proteins with streptavidin coated beads. And then you can do a downstream omics analysis. So first we want to look at mitochondrial, right? So whether we can tag mitochondrial with this proximal labeling enzyme. Because we're mostly interested in the complex, OXPHOS complex. So we used a bait protein called SP2 or STOM2 protein. So this protein lives inside this inner mitochondrial membrane and facing the matrix. That's where the OXPHOS complex actually lives and generate ATP from this inner mitochondrial membrane. So we tag the enzyme to this bait protein, and then we can activate this enzyme by adding biotin to the cell culture for half an hour. And then we can lyse the cell and then enrich the biotinylated proteins, look at those proteins. When we do a go-term analysis, we found that indeed we're enriching a lot of mitochondrial proteins from the cell, and especially we're enriching mitochondrial matrix proteins and inner mitochondrial membranes, which is where we're targeting at a 10 nanometer labeling radius. We can, when we really see any mitochondrial outer membrane proteins, which really demonstrate that the specificity and the spatial resolution of this approach. And when you look at the protein interactions, you can see this is our bait protein. So we can pull down both the direct interactions to those proteins and also those transient interactions, and also the proteins that are just generally located at the matrix and in their mitochondrial membrane. So those are the typical respiratory chain complexes. Those are the OXPHOS complexes we're really interested in, mitochondrial ribosomes and mitoprotein processes within the cell. So in using this way, we can actually specifically pull down those mitochondrial proteins, especially inner membrane mitochondrial proteins and matrix mitochondrial proteins, without the big background of cellular molecules and outer mitochondrial membrane. So we also look at the lysosomes, since we're also interested in lysosomes. So for the lysosome, we tag the lysosome outside of the lysosome. The reason we do that is really hard to tag inside the lysosome because lysosome is acidic. So most of the proteins or enzyme, especially if you tag something, it's not endogenously leaving inside the lysosome, it's pretty much going to get cleaved off, degraded inside the lysosome. So we tag the lysosome facing outside of the lysosome to look at how lysosomes interact with the cellular environment. This is also quite interesting because lysosome fuse with other organelles all the time, and then because it tries to every time during the autophagy, it engulfs with the autophagosome that eated all of the trashes and the misfolded proteins, and the lysosome is going to fuse that. So what's actually most interesting is the outside of lysosome and the membrane of the lysosome to look at the dynamic lysosome activity. And this project is led by Ashley in my lab, and Ashley is actually the first PhD student in my lab. Okay. So this, we first wanted to validate whether it works. So when we do strobavidin containing SA stands for strobavidin, it marks the biotinylated markers. So anything that's biotinylated surrounding this proximal labeling enzyme is going to be labeled. So we see that those are the signals of the biotinylated signals. When we put that together with LAMP1, which is our bait protein, and this LAMP1 is a classical lysosome marker that lives on the lysosome membrane. So when we see the LAMP1 actually co-localize with the strobavidin, actually very good, around this cell, and this is a cell nucleus. So of course, lysosome is going to leave outside of the nucleus. So you see that they co-localize really well, really demonstrate that the location of this enzyme is working, and the enzyme can actually be activated by this 1 minute of H2O2 treatment and half an hour of phenobarbital treatment. So when we look at what kind of proteins we can pull down, we see, of course, the LAMP1, which is our bait protein, and all the known endolysosomal membrane proteins that we can pull down, and ATPases. Those proteins leaves on the lysosome membrane, and they're actually key factors to control the acidifications of the lysosome. So study have found that if there are mutations on the ATPases, a lot of time is lethal, because if the lysosome can't really function, then it's really detrimental to the entire organism. So we're not only able to look at to identify and quantify those proteins that stably interact with the lysosome and lysosome membrane, we can also capture those transient interacting proteins. Those are the membrane trafficking proteins. So for example, this SAC proteins and annexin proteins, those proteins are actually in charge of how the lysosome traffics, especially along the neuron. And also control, we also see some autophagy proteins. So that means the lysosome is engulfing with and fusing with the autophagosome. So as we are developing this tool, so a lot of the projects in my lab right now is in the stage of method development, because we are a relatively young lab. So when we try to develop those approach, now it's working really well. So we have a lysosome proximal labeling probe that we can look at the lysosome microenvironment. We also have a mitochondrial probe that look at inside the mitochondrial environment. But we also faces a lot of technical challenges in this proximal labeling field. There are a few ones I'm going to list it out, and I'll tell you how we can actually to tackle those challenges. So when we look at proximal labeling mass spec, this is one of the example where we look at LAMP1, which is our bait protein, and also the highly abundant other proteins in the cells that we enriched. There are a group of proteins always show up all the time. Those are PCCA, PCCB in actually all three different LAMP1 apex cell lines. So those represents that we're trying to tune the different expression levels of this beta protein. So we see both the knock-in line, the overexpression lines, all have really abundant those are carboxylases. And even in the NES, which stands for nucleus exporting signal, so those are the control cell lines that we use that labels pretty much every proteins inside the in a cytosol. So it serves as a spatial control compared to the LAMP1. So both NES and the LAMP1 apex have high expression of those high levels of those carboxylases, where no apex, even no apex control, we see them. That's because for no apex control, this is the cell line that we didn't incorporate any enzyme, but went through the same treatment of Biotin, adding half an hour of Biotin. So then we took a closer look at this. Those are the endogenous Biotinylated carboxylases that lives inside the mitochondria. So those proteins itself is a Biotin-dependent carboxylases. So you can imagine when you add Biotin trying to enrich your enzyme at a different location, those carboxylases also eat up your Biotin, and then they're really happy and they just keep growing, and then you have a lot of those proteins. So those proteins is really hard to get rid of. So we have to think about a way how we can actually utilize or get rid of or at least reduce the levels of endogenous proteins. So when we look at, although we can't really get rid of it, we found that especially the highly abundant three PCCA, PCCB, and PC, they have a pretty consistent expression level in different treatment. So we can actually use this as a normalization. So for approximate labeling experiments, every time we use just a 1 minute treatment of H2O2, so there's quite a variations happening from batch to batch. So when you look at a different batch labeling of the proximal labeling and comparing their protein intensities, and if you look at the same batch where we just do those at the same time, it's much tightly clustered. But if we can normalize that to the PCCA, which respond to the variations where you add the Biotin and H2O2 treatment in a cell, right? So you really can actually cluster them back. So especially for the different batches of cells. So this really enabled us to compare different batches of the proximal labelings using this PCCA normalization. So in the same batch also is still pretty good after normalization. So that's endogenous Biotinylated proteins. I'm gonna come back to this topic again, but let's look at another challenge first, which is the strobabidin contamination. So when we try to enrich protein Biotinylated molecules surround this protein enzyme, we're using a strobabidin-coated beads, and strobabidin and Biotin have a really strong interactions. Actually one of the strongest non-covalent interactions in nature. So you can imagine it's a very specific enrichment. So only the Biotinylated proteins usually can enrich, and you do very harsh washes, and because of the strong interaction, they won't be washed away. Only the non-specific molecules will be washed away. But there's also a problem because now it's enriched on the beads. It's really hard to get them off. It's really hard to elute them because they just bind so strongly. When we look at, so we first want to evaluate how bad is this background signal. So what we did is we just using the strobabidin beads and then put enzymes, trypsins directly on the beads. So that's the typical approach that people nowadays for proximal labeling mass spec, because you can't really elute them. So we just add enzyme directly to the beads. So when we cut those proteins that links to the Biotinylated proteins, you also cut strobabidin. Strobabidin is actually a polypeptide. So there are a lot of strobabidin signals, especially even when we digest empty beads. So you see those little red dots are all strobabidin signals. And of course, you're gonna see keratins because we always see keratins from our skins falling off from the hair, right, it's everywhere. And we also see trypsins as sort of a background signal with a blank sample. So what we wanted to see if whether we can actually elude this, think of a way to reduce the strobabidin signals. So that's one of the way we trying to approach this is to design a cleavable Biotin method. Cleavable Biotins, for example, this SS Biotin where you put an SS bond in the Biotin molecule has been actually used for a lot of chemical cross-linking mass spectrometries, but it hasn't been used for proximally labeling mass spec. So Haorong, a graduate student in my lab, are trying to evaluate whether we can actually use this style cleavable Biotin in the proximal labeling experiments to so that we can actually just cleave this disulfide bond after enrichment. So as normal, we'll just conduct the same reaction, and instead of Biotin, we're going to add SS Biotin into the cell culture. And the SS Biotin is going to bind to the proteins nearby this the enzyme, and that can be enriched by strobabidin. But then we don't have to digest on bees, we can just cleave them off. After we cleave them off, then we can digest those proteins without touching the beads. We can just get rid of the beads, right? So this really helps us reduce a lot of the strobabidin signals. But I wanted to mention that even with this, we still see a little bit of strobabidin signals from the sample. I think just because some of the strobabidin binded probably falls off during the washes. So it's really hard to completely remove it. But if you look at this dramatic signals, now they're gone. So you can't really, they're definitely not mocking your other proteins. So with that, we can dramatically reduce the strobabidin signals. And we first look at whether we can make this reaction complete. As a chemist, when we look at a chemical reactions, we really wanted to push the equation to the right end. Right. So when we look at, so we first evaluated the Biotinylation with a chemical labeling, where we use NHS, SS Biotin, and NHS normal Biotin to label just a normal cell lysate. So in this way, we can, NHS labels the amine group. So it is sort of mimic the turbo ID enzymatic reactions in the cell. So when we do this in vitro first, we can label the protein with this NHS, an SS Biotin or NHS Biotin first. Then we can add TCEP or DTT, which is reducing agent, to cut this disulfide bond. So speaking of that, I really recommend using TCEP over DTT because it's much stable and it doesn't have this very bad smell compared to DTT. So when we use TCEP to cut this disulfide bond, we also need to cap it. Otherwise, it might reform again. So we add IAA to react with the end of the disulfide bond, so we can add a carbomethylation group onto this disulfide bond end so that we can cap this, prevent it from reforming again. So we did quite some optimizations in trying to get an optimized concentration of this TCEP9A so that we can get a complete reaction or nearly complete reaction for this chemical labeling first, and then we move on. Then we use the same concentration for the in vivo proximal labeling approach. So another sort of bonus that we can get when we're trying to get rid of strobabidin is we can actually also see Biotin-related site of those peptides. So you can imagine when we use the beads to do an on-beads digestion, the actual Biotin-related peptides is actually going to remain on the beads. What you digested is actually the non-Biotin-related peptides. So you actually lose where those proteins can actually bind to the Biotin, which could provide structural information of protein folding. So when we use this cleavable Biotin, because we can cleave it entirely of those proteins, we actually are able to get a lot of Biotin-related peptides especially using this chemical Biotinylation. And traditionally, we think that it only labels at amine group that is mostly the lysine, because lysine has a side chain amine, so that amine can be labeled, and the other amine is linked in the peptide bond, right? So when we look at the different Biotinylation site, we actually found that it actually can label also other amino acids, although not as abundant as lysine for both SS Biotin and Biotin, but serine at least some of the serine can also be labeled. So with that, I actually recommend that in the future for doing biotinylation analysis, we wanted to tell the software to not only look at lysine biotinylation, but at least look at serine or threonine biotinylations, which could be a part of this biotinylation pool for where the peptide links to biotin. So because it binds to lysine, and we often use trypsin to digest proteins, which trypsin cleaves at lysine and arginine site. So we had this question whether this biotinylation at lysine could reduces the trypsin digestion efficiency. So in fact, it did. So when we use the unlabeled protein, which doesn't have any biotinylation, it's just a normal protein lysate, majority of the protein have 0 miscleavages. So that means the trypsin cuts really well. It cut most of the K and R. But when we look at biotin labeled proteins, majority of the proteins actually have at least 1 miscleavage. And then we look at that and mostly almost all happened at the K lysine site. When we look at SS biotin labeled proteins, it's the same trend. You still have majority of them have 1 mislabeled 1, but you do have a little bit less of 2 miscleavage or 3 miscleavage. This is probably because that when you cut and cleave this SS biotin, your proteins are so the biotin, the little residual chemicals that links to protein is a little smaller, so it might reduces some of the miscleavage, but still you compare to the unlabeled one, it's a huge miscleavage problems. So this is especially problematic for on-bees digestion, because for on-bees digestion, you're gonna collect the supernatant. So if you don't cleave it efficiently, you won't see it. But for our assay, so because we elute them completely, so even if we have some miscleavages, we can actually loosen up the threshold for proteomics analysis to tell the software to set a 3 miscleavage or 4 miscleavage instead of we routinely set as 2 miscleavages. So we can actually see those proteins, miscleaved proteins, if we can elute them using the cleavable biotin method. Yeah, so that method is recently published and featured on the cover, and really due to the hard work of 2 of my PhD students working on this. So then I'm gonna switch gear to the last project I'm gonna talk about is the look at how cells homogeneous and protein turnover in neurons. So we're looking at neurodegenerative disease. So one of the model, that direct model would be neurons, right? So mouse, traditionally people use mouse neurons because it's easy to get, and you can't really take neurons from human brain, and even if you get like cadaver, and then the neurons is mostly dead, so you can't really do that. But in recent years, this human iPSC-derived neuron platforms has been developed, and especially this is what I actually learned during my postdoc research, where you can actually take a skin cell, some skin cell from human body in patient, and then turn that skin cell back to stem cell, and then you can derive the stem cell into neurons. So this is actually invented by a Japanese scientist, but what we are trying to improve when I was a postdoc is speed up this process. So most of the traditional way to derive iPSCs into neurons takes about 2 to 3 months, where if you want to do omics, you don't really have, you can't really generate as much materials for different omics analysis. So my postdoc advisor developed this approach to incorporate transcription factors into the stem cell that can drive the stem cell to a pure cortical neurons, so that you can actually get pure neurons in 2 weeks from stem cell. So using this approach, we want to look at how this protein turnovers in neurons. And the way we're doing this is by dynamic SILAC, where you can culture everything just in normal media. When the neurons reach maturation or at around day 10, you can switch the media to the heavy lysine containing media. And then you can imagine all of the heavy proteins are started the synthesis. So the new synthesis happens in the heavy lysine, where the old proteins continues to degrade. So if you take multiple time point after this median switch, you can actually generate the protein degradation curve and synthesis curve and find out where they intercept. And that is your half-life of the proteins that you can look at. And if we use proteomics approach to couple with this dynamic SILAC, we can look at the global protein turnovers in neurons. So this is, you can see an example of some protein degradation curve and synthesis curve at different time point. And this is an example of some key lysosomal and neurodegenerative related proteins. This is a lysosome enzyme of having a actually very long half-life, 5 days. TDP43 is one of them, but actually a marker for FTD and neurodegenerative disease. A lot of the TDP43, especially false-related TDP43, are started to aggregate in neurodegenerative disease neurons and tau proteins as well. So you see that you can actually measure those half-lives, and then you can look at whether those half-lives can be influenced by genetic mutation. So first, I want to look at the wild-type neuron, where the neuron is pretty healthy, and then we have a median half-life of 4.8 days. So this is much longer than like a cancer cell or other type of skin cells. So, but if you do this experiment in a mouse, it's actually even longer. But for brain, you can sort of imagine, because we need a lot of neurons or long-lived proteins so that we can keep long-term memory. Right, so that actually makes sense. A lot of proteins have a really slow turnover, and actually one of the lowest, longest protein lived is, I think this is like even 60 days around. That what we see in the neurons, and this is actually a protein that autophagy machinery. So the proteins that get degraded usually turns out faster, but the proteins in charge of degrading other proteins are turning out actually very slow. The histone proteins and nuclear proteins belongs to the nucleus and nuclear pores usually have really slow turnover, where proteins related to neurosecretory or neurotransmitter regulations or proteins related to the ubiquitin pathways that quickly always tag other proteins for degradation have a very fast turnover. So then we want to look at genetic mutations. One of the disease we're really interested in frontotemporal dementia, which affects the frontotemporal lobe of the patient. So one of the major causes of frontotemporal dementia is this granulin gene. So when the granulin gene is mutated either in the family or through sporadic mechanisms, it's going to influence the progranulin, which is a protein that lives inside the lysosome. All of these granulin mutations are loss of function. What does it mean? It means if you have any kinds of mutations in the granulin gene, it downregulates the progranulin. So when there is mutation, the progranulin actually have much less progranulins expressed in the lysosome. That of course makes lysosome not happy, but how is the specific functions or detailed molecular processes that are influenced by progranulin deficiency and granulin mutations are not well understood. So what we wanted to use is we wanted to use our proximal labeling lysosome approach. We wanted to use the lysosome dynamic SILAC to measure the protein half-lives that are influenced by this granulin mutation in the neurons. So we can actually grow the patient fibroblast with the granulin mutations into cortical neurons. We can also get, we can actually correct this mutation in the IPSC stage to generate an isogenic control for this patient, for individual patient line. And then we can capture the dynamic lysosome with proximal labeling. So we have wild type and also the granulin deficient neurons. You can also, so there are two ways you can generate this model. One is to use a use a patient line that have the mutation. Another, if you start with a healthy line, you can actually knock out progranulins in the healthy line and look at the comparison. So when we look at the LAMP1 apex, which is the lysosome microenvironment in those neurons, we see that proteins that are calcium dependent and hydrogen exporting ADPases actually are moving away from the lysosome or reduced around the lysosome, which could make sense because we actually did found that progranulin deficient neurons the lysosome pH is abnormal. So the both calcium dependent, now those ATPases, as I mentioned before, actually controls the lysosome acidification. Where when we look at the upregulated proteins in this data set, those are the proteins that are especially lysosomal enzymes. So those enzymes that are trying to degrade proteins are increasing, and then enrich, especially endosome to lysosome transport is increasing. So this is probably due to the deficiency of progranulin, which is a protein living inside the lysosome. And because of the deficiency of progranulin, the lysosome is trying really hard to synthesize more enzymes to compensate for this loss of the progranulins inside the lysosome. So now we are trying to expand this workflow to a mouse model. So this is in collaboration with Sadia, MD PhD students, and also in Dr. Michael Ward lab at NIH, where we are trying to expand our lysosome protein labeling approach to label fixed mouse brain slices. So now this work is ongoing, where we can actually see that in our preliminary data, the strababidin stained signals, those are the biotinylated markers, do co-localize with LAMP1 in the mouse brain. It's not as clear because you have a lot of background signals in the mouse brain tissue slice, much more complex environment than actually a neuron culture. And when we also look at the protein turnovers of IPSC neurons in the wild type versus the progranulin deficient neurons, we found that loss of progranulin really impairs the lysosome degradation, because we see a lot of lysosomal enzymes that have a either a faster turnover or a slower turnover, and proteins that are actually very directly related to the frontotemporal dementia. These progranulin deficiency causes to the final stage of the neurodegeneration also have a changed turnover, either faster or slower. So now we're trying to, this is ongoing work, so we're trying to combine those different data sets and to try to understand and also inhibit the lysosome specifically and see how those proteins may change, and whether those proteins turnover changes are directly related to the lysosome dysfunction. And this is a go term when we see that a lot of protein changes are actually directly related to what we wanted to see with the frontotemporal dementia and other neurodegenerative disease. So you can actually see neurodegenerations, although it happens in different part of the brain, they really share a lot of common mechanisms. So by studying mitochondrial and lysosome, really hope to decipher some of the common mechanisms that leads to different type of neurodegenerative disease. So with that, I would like to conclude. I'm hoping that shared with you how we can actually develop mass spectrometry-based technologies to not only look at the global proteins and small molecules, but also with a very good spatial resolution to look at approximate labeling mass spectrometry and address some of the technical challenges belong to that method, and then applying that to try to understand neurodegeneration, both the lysosome dysfunction and mitochondrial dysfunction. Yeah, with that, I'd like to thank my my lab. And this is actually a recent picture where students wearing masks in the lab, and this is actually where we very close to where the students are sitting, and our pressures mass spec in the lab, which generated all the data for this, and also our collaborators and fundings. And also we're hiring, so we're hiring actually at all different levels, so graduate student, master's student, PhD, postdoc. So if you know anyone wanted to come to the DC area, we are actually in a very nice building, only I think less than 10 years old. So definitely recommend that my lab to them. Okay, with that, I'm happy to take questions.
Thank you so much, Dr. Howard. It was really enlightening, and also I think the most proteomics we have ever had in our holy halls here. All right, so Arpana, why don't you take it from here?
Great, thank you, Dr. Howe, for such an enlightening talk. Like Oliver said, I did learn a lot of proteomics, and I have a couple questions on that. Starting with the biomarker discovery on MELAS, there's a couple questions regarding that. For proteomics of the OXPHOS mitocomplexes. Your p-value was less than 0.05 without FDR. How many samples did you use? And could it be that the apparent upregulation or downregulation was just random and not significant? Maybe because of the low number, the low number of samples?
Yeah, that's a great question. Before I answer that, I wanted to address to Oliver's question about because I love metabolomics. And now, especially when I just started the lab, the first thing we do is to buy all those metabolite standards. But as a new lab, we have limited people. So my students are gradually, so now we have built this IPSC-neuron model in the lab. We really wanted to get more metabolomics data. So that's our ongoing efforts. So hopefully, maybe next time when I see you in conference, I'll have more small molecule data to show you. Yeah, so it's a great question of the FDR. What we found that especially, I think is really true that if you don't have enough replicates, when you do FDR correction, a lot of them get cut off. And I think there are two reasons. One, it could be a random, it's not a true change. Another is actually just because it's not enough replicates. So when we do the proteomics, we did use FDR cutoff for the both proteomics metabolomic hits. But when we try to merge the different data sets, so for proteins, we have DIA and DDA data set. For metabolite, we have all the different like polar metabolites, non-polar metabolites, HILIC and RP. It's really hard to merge only the FDR corrected small molecules. We only have very little left at the end. So that's why we actually use just the p-value instead for the final results. But I think it's definitely correct that I think it definitely needs to be validated by more replicates. And for a patient fibroblast, it's really hard to grow them in a lot of amount, especially those patients are not healthy fibroblasts. So we used 4 biological replicates. But I think we definitely in the future, I think instead of doing more fibroblasts from the same patient, if we can get more patients, I think that's a better validation. Yeah, it definitely needs validation.
Great, thank you. So referring to, in sticking to that same section in biomarker discovery, referring to your finding on reduced arginine and arginine biosynthesis, what method was used to quantify the significance of the pathway? I believe it was on slide 12.
Yeah, so that is just based on how many proteins you identify from the pathway that are changing, or how many small molecules that are changing, and merge those significant proteins and small molecules into the joint pathway, and to find out whether these pathways are changing. And then you want to plot them together and see whether it is changing. And also we're very interested in this arginine biosynthesis because patients are taking arginine, but it's not enough apparently.
I see. Going back to, talking about validation studies, in your larger patient cohort validation study, will you also use fibroblast as samples?
Yeah, so we have to use fibroblast for mitochondrial disease, because if you do urine, blood, or CSF, you don't have mitochondria in those bowel fluid, then it's really hard to actually look at directly.
And how easy it is to, I mean, sorry, I'm not a clinician, how easy it is to just scrape off from epidermis, or how do you get the connective tissues, or what is it?
Actually,
how do you get the fibroblasts?
Actually, don't know how they get the fibroblasts. Based on our collaborator, I think it's pretty easy to get the dermal skins from the patients. The hard part is how many of them leave in a dish. So they actually get a lot of the dermal fibroblasts from the patient and put them in a dish, and then select, and then do more selections. So it's actually seems like it's not very hard, just based on what I heard.
Zach, do you know?
I don't know how they obtain them, but I know they're really hard to grow. I've tried growing them, and they just, unless you have like a really rich media or other cell types, I mean, you can maybe get 1 million cells, but they're tough to grow.
Yeah,
Okay.
that's why we had to, yeah.
Yeah, we usually get plasma, and I really am not fond of plasma. And then we get plasma data, and people talk about mitochondria, and I get really fuzzy in my head. right? Because they, it's like, okay, you have plasma, for God's sake, not even cells. And yeah, you see all these papers talking about pathways like glycolysis and so on. Like, no, happens in cells, not in plasma, right? So it's but if it's really that hard, if you could talk to your collaborator and see, if they want to have comments on that, I'd be really curious, because I also need to educate my collaborators so that they don't give me plasma, because I don't. In terms of metabolism, I'm really not fond of plasma, honestly. And it's like 60% of the samples we have in the lab, I guess.
Yeah,
all right.
Sure. Let me ask my collaborator and more details about how they actually grow and collect that.
And just a follow-up question. Sorry, Arpana, if I could.
It's okay.
So you then said you can also like convince the fibroblasts, if I understood it right, to convince them to become pluripotent again, like induce them to become pluripotent, and then you derive neuronal cells from them. Is that correct?
Yeah.
Okay. So I got this right. Are these neuronal cells similar to an old brain? I mean, people develop these diseases when they get old, right? So you have fresh cells, just like, like really fresh. Would they see the same issues?
Yeah, that's a great question. I think it's not going to mimic the aging process.
Yeah.
So, and it's going to be really hard to age the neurons in a dish.
Yeah, right.
So that's why I think, so the approach we are looking at is the mutation. So if you are trying to look at what does mutation do to neurons, then it's a really good platform. But if you're trying to look at what age does to the neurons, then I think you almost have to go to mouse models. So you can actually get mouse neurons directly from mouse embryonic cells, but it's a pretty young, it's a young neuron. You can't really get, or I guess different ages of the brain directly. So that would be the more of a mouse model. So we can't really mimic the aging process. Yeah.
We've just published a database in a paper on the aging mouse brain. If you're curious, just look it up.
Yeah, sure. I'll look it up, definitely.
But we didn't have any diseases with it. So just like as an atlas. All right.
Great. There's a couple questions on the methods that you used for metabolomics and proteomics integration. I was curious about how much fibroblasts you ended up using. You said it's very hard to, collect that sample. And then what would you recommend as a minimum amount if someone was interested in doing, something similar?
Yes, Zach was mentioned, it's really hard to get even like 1 million. So we did 4 replicates. Each replicate is about 1/3 of a million. So the total is about a little over 1 million. Yeah.
Okay, great. There was a question on what the pooled samples were and how it helped in the data analysis.
Yeah, so this is actually, I think it's definitely, I really recommend whenever you can to generate a pooled sample, because you take a little bit from each sample. So you don't really cause any sample loss for individual sample, but you have a pooled sample, then you can use it to not only to monitor the instrument system, the instrument's stability, but we mostly use it to identify metabolites. So we do a bunch of MS on those and MS1 on the pooled sample, so that we can match that to our library and also the online library. And then you put those data to import those data together with the biological comparison data, where we just did full MS on those. So you don't lose the instrument time for those full MS scans.
Right, I agree with that. I mean, we also use pooled samples, and one thing that's really helpful is when your sample type isn't available as a standard reference material. Using a pooled sample can help in that regards too. One last question on the biomarker discovery part, referring to your multi-omics integration. How did you integrate the two data sets, or was it just inference from one set to another? You know, you found the metabolites and then looked for the proteins, or vice versa, or did you actually integrate in order to, you know, report the findings that you did?
Yeah, we use Metaboanalyst. So I think a lot of you guys are probably very familiar with that tool. So we have a list of changed proteins, and we have a list of changed small molecules. And then we put those lists together along with the ratios together into the Metaboanalyst software. And that's going to generate the multiple like pathways that are dysregulating. But that's actually just the first step, because a lot of the small molecules that are not reaching statistical significance might also be interesting, because it might not reach statistical significance just because you don't have enough replicates. So what we have been doing is we first use the significantly changed hits to generate this, the pathway of interest that's stood out in the multi-pathway analysis. Then we draw this pathway, specifically pathways, and we try to put all of the identified molecules in this specific pathway. So you first choose maybe two or three or five pathways, and then you try to put all of the molecules that are identified to this pathway. And then you can categorize them into upregulated, significant, upregulated, not significant, downregulated, significant, downregulated, not significant, so that you have actually a more complete picture of what's going on. And that also needs to pair up with the biological knowledge about, you sort of know that what kind of pathways you're expecting to see. So that needs to be paired up together.
Okay. Yeah, it's always interesting to learn how people actually do it, you know, so I really appreciate that. I'll give it a try myself one of these days. Switching to the organelle proximity labeling studies, have you tried in situ mitochondria labeling and see if there's a difference in the mito counts?
What is mito count?
Mitochondria counts.
Okay, so we do add the so the label is in situ, actually in the cell. That happens when the cells is still alive. The mitochondria get tagged, but we can't really see how many mitochondrials they have. We just see what are how many proteins that belongs to the mitochondria. So we assume that so right now the in situ mitochondria is still in method development stage. So we didn't actually compare it with a diseased mitochondrial or healthy mitochondrial. So in the method development, we just see the protein identification. So we can't really get a count of a mitochondrial count. So what happens in this MELAS patient mitochondrial, I think it's not the total amount of mitochondrial, is the unhealthy mitochondrial, the percentage of the unhealthy mitochondrial in the total amount of the mitochondrial.
I see. I have a question, and maybe it's a little general in my lack of understanding of proteomics, but can you please explain how miscleavages affects your proteomics data and like what challenges you face when you're processing the data? My understanding, it's like a mistake, right? So like, how does that all affect, you know, down the path workflow? Or
Sure. So you imagine like a protein, right? It's really long peptides, and then you add an enzyme so you can cut it at a specific place. The most common used enzyme is trypsin, which cut at arginine and lysine. So we know that there's never a chemical or enzymatic reaction that's 100% efficient. So you have to add enough enzyme so that it can cut it really well, but sometimes it also missed it. So for example, if you cut it at if it's 100% cleavage efficiency, then all of your peptide only had 1 R or 1 K at the end of the peptide. That's where it cut, right? So if you see 2 arginine in the peptide you identify, that must means that one of the arginine was missed. So that's miscleavage. So this is So we wanted to add enough enzymes so that it reduces that miscleavage, but it's actually inevitable. So you always see some kind of miscleavage, sometimes because the enzyme, sometimes just because of there might be something that blocked the enzyme, or if there's a proline actually linked to by the lysine is going to protect the lysine from cleavage. So in the normal experiment, you always see a little bit of miscleavage, but you should see majority of them have like zero miscleavage.
So do miscleavages affect like the accuracy of proteomics data or identification in proteomics?
So I think it's mostly identification. So if you because during identification there is a false discovery rate, right? So you tell the instrument to look for arginine ending or lysine ending proteins. So you're most confident. If you have a peptide that have so many miscleavages, there is possible that it is a miscleavage, but it could also be a misidentification. So that's why we want all of the peptide to have zero miscleavage, so that you are most confident.
Okay, great.
But actually, it does. So the nowadays the fermentation pattern really provide a lot of information. So even the miscleavage peptides, you still see a lot of good identification. In terms of proximal labeling, the major downside is if you try to cut it on beads, right? It's on beads digestion. If you don't cut it, it won't come off and you won't see it. But in solution, it's actually fine.
Okay, great. Thanks for enlightening me about more proteomics. So the last section of your talk on dynamic protein turnover, how feasible is transferring this methodology to different cell types like microglia? Could you co-culture the neurons and microglia or use brain organoid models to understand better how these cell types interact regarding lysosomal activity?
Yeah, that's a great question. That's actually something we wanted to work on, but we don't have that bandwidth yet, and we don't know how to culture organelle yet. But a lot of labs actually does microglia culture and organelle culture. It's actually a very quite recent development in recent years. But in order to do the half-life measurement, those needs to be that you can actually change them into heavy. So if you are trying to get the cells, get the microglia or organelle directly from mouse, then you have to feed the entire mouse into heavy, which is very expensive. But the cells, one of the challenge we found is you almost have to talk to the company to get customized media. Because right now for SILAC, you can buy SILAC media for cancer cells, like very easy. But for neuron culture or microglia culture, people are using different media, and then we're still improving the media and the culturing environment. And that sometimes can be difficult to turn that all into heavy.
Great, thank you. I'd like to open it up to further discussion. We definitely have more time. That's all the questions in the chat, but feel free to continue to ask questions or just go ahead and unmute yourself if you have a question for Dr. Howe.
Maybe Arpana, you can already bring up the next speaker.
Oh yeah, I can do that. So I guess with that, thank you, Dr. Howe, for answering all those questions and for giving a great talk. I definitely learned a lot about multi-omics. I'm always very curious about that.
Thank you again, Ling Hao. Hope to see you again soonish. Good luck with your work. Right?
Thank you. Thank you for the invitation. I already asked my student to register for your seminar. So
all right.
Okay.
Very good.
Thank you.
Thank
Bye.
you.
Okay,
Bye-bye.
bye-bye.